almost all of us here will know somebody whose life has been affected by cancer. And in fact, according to Cancer Research UK, nearly half of us will develop the disease at some point in our lifetime. But thanks to decades of research, by the likes of the people that we've got on stage here, survival rates have doubled in the last 50 years. And today we're looking at the next wave of innovation that could save even more lives. And we've got two groups of scientists on the stage today. One is developing an incredibly precise laser-driven particle beam, and the other is using tiny magnetic particles called nanomaterials, and they're so tiny, aren't they? You can fit thousands of them onto one human hair, and they're using these materials to target cancer from within. And the whole idea of all of this is ultimately to make treatments for cancer more precise, more effective, and crucially, as anyone who's gone through treatment or knows somebody who's gone through treatment will know, just to make it kinder to patients. So let me introduce our panel first. You've got Professor Kenneth Long, who's a Professor of Experimental Particle Physics at Imperial College London, as well as the Science and Technology Facilities Council. And Professor Pat Price, next to him, is an academic clinical oncologist specialising in advanced radiotherapy and molecular imaging at Imperial College London. And joining them is Professor Thanh, to my right. He's a Professor of Nanomaterials at University College London, and Professor Martin Forster, who is a consultant medical oncologist, also at UCLH. So we're going to start with a couple of quite short presentations just to kind of get us across what this work is all about. Could you guys start with just telling us a bit about the current treatments that's kind of relevant to what you're doing, radiotherapy, what that is, how it works, and how this is different?
So, of course. So, cancer, right? It's basically our own cells that have gone out of control. You can imagine if you scratch yourself, you'd form a nice scab and then it would heal. But cancer, the DNA doesn't stop, and it grows and grows and grows to be a tumour, and then it can spread. So it's all about the cells of our own body misbehaving, okay? So when you get a tumour, there are three main ways to treat it. If it's just localised, great. You can cut it out. If it's on the top of your arm, just cut it out. Or you can actually irradiate it, which means firing X-rays at it to ablate it, to make it all go away. Or the third way is to give it drug treatment, and that's various drugs, hormones, immunotherapy, whatever. They go all around the body, and then they can affect the tumour. So they're your three types, okay? So we're looking at radiotherapy. Radiotherapy is needed in half of all cancer patients at some time, and it's involved in 40% of cures. So 4 out of 10 people with cancer, if you're going to be cured, you need radiotherapy. Big ones are things like, look, we're all talking about prostate cancer at the moment. The treatments of that are either surgery or radiotherapy, lung cancer, this sort of thing. So it's really important. Now, what radiotherapy does, it's basically X-rays. So, you know, if you have an X-ray, if you've broken your arm or something, the X-rays goes through and then you can see it. Well, these are really high-powered X-rays. They actually are really high energy. Now, we found those originally. Radiotherapy was started Marie Curie's day when she found radium, gave off, and he thought, oh, that cures cancer. Oh, that's very interesting. And so over the years, we've been trying to work out, how's the best way to use this? So that's how modern radiotherapy is. And it's so good now, we can focus it down right on tiny little things. But there's more excitement also in terms of not just X-rays, but particles. So particles have energy. So you may have heard of protons, they're a particle. And what's good about them, X-rays go through the body and obviously go out the other side, whereas particles screech to a halt, it's called a Bragg peak, and deposit their energy. So that's really good if you want to focus it like children and that sort of thing. But even more exciting, there's high energy particles like carbons and helium that Ken will talk a lot, in CERN you've got antimatter, all these things that are really, really exciting. And there's evidence that they probably damage the DNA. We want to damage that DNA that's making the cells grow. They damage it probably far better, kill far more precisely, and also affect other things like the immune response coming in. So you get a double whammy. So these could be great, so that'll be another big armamentarium. Trouble is, at the moment, you need these massive big accelerator colliders, CERN is under Switzerland swinging it round, other places great round. That's not compatible with the hospital. So, smart idea, lasers to produce the particles, to then focus them down, and then treat the patients with that. So hopefully we'll get a machine you could buy and that sort of thing. And it's really important. Radiotherapy, we don't all get radiotherapy. 50% should have it. In the UK, only 35% get this. There are 38 countries in the world who have no radiotherapy. Not that it's expensive, it just needs people and kits. That's what you like. So Ken is going to tell us this new way. It won't replace normal, it'll be addition to it.
Yes. So what we want to do is to take a high-power pulsed laser to create the proton and ion beams. The advantages are that you make a higher energy protons than conventional sources, and you make them in very short bunches, so you get very intense, instantaneous bursts of radiation. The advantage of the laser is when you shoot the laser, a known time later, the particles arrive at their target. That means that you can tune the time between the particles arriving, and that means you can really explore first the science of how the therapeutic effect is generated by the fundamental biological processes that are induced by the radiation, and later you can really tailor the treatment to the individual patient and the individual disease. So probably everybody in this room is a different size and shape, and the tissues are in detail different inside everybody's body. With modern detection systems and modern computing systems, we want to develop a facility which would be able to measure the people as they walk into the room, identify where the tissues are, and have all of that interlinked with the laser-driven accelerator so that you can really shoot when the stars are aligned and thereby reduce the size, the investment cost, and the dwell time, so that how long it takes to treat the patient. Because of the effects that Pat mentioned, in principle, these techniques can reduce toxicity, and the speed means that we can reduce the discomfort, or the length of time for which the patient is discomfort. So we really want to make patient benefit in principle with better treatment, and in practice with more comfortable techniques.
Okay, so super promising stuff happening there, and very exciting. Look forward to getting into the meat of that. Let me turn now to you, Professor Thang. Yes, we'll put your slide on so we can have a look at what you're doing. So you're taking a very different approach, aren't you? Can you talk us through this, the concept behind this magnetic hypothermia, and how these tiny sort of nanoparticles are used to kill cancer?
Right. So when you have cancer, I'm talking about like solid tumor, and the way you can treat the cancer locally is that
we just need to grab your slide, don't we? Is there...
Yes, and next.
Oh, there we are. Excellent.
Yeah, you introduced us already, so the next one, please.
Perfect.
Yes. So if you go down to our stand, you will see that we have this, I call it like, if you imagine this gingerbread man, but actually silicon. And we inject here these magnetic materials in very much like the tumor. So sadly, if somebody have a brain tumor, then you inject the nanoparticle inside, and you're going to treat it locally. And so how does it work? As you can see from, too fast, sorry. If you can see there that the device is very, very small, like your hand. And you place locally, and it's going to hit locally only the magnetic nanoparticle. So it doesn't affect any other area. So you treat the cancer locally. Why? Because the magnetic nanoparticle can interact with alternating magnetic field, and they're going to change the spin. And the field not need to be very strong. It's only 200 times more than the Earth magnetic field, but the frequency is quite high. It could go from 100 to 500 kilohertz. And then you're going to hit the nanoparticle locally, and why? You can kill the cancer cells, because the cancer cells are more susceptible to heat, so you only need to heat up between 39 to 45 degrees, and you can causing apoptosis and necrosis. That's the process of killing the cancer cell. Can I have the next one, please? Yes, so you realize on the nanoparticles that you are using to treat the cancer. So you have to make them hitting very well. So currently in phase 3 clinical trial by McForce on the glioblastoma, they use the nanoparticle, which is iron oxide, nothing more than the iron rust on your bicycle in the shed, but they're hitting not very well. So we invented the technology that we can make the nanoflowers and the interaction between the cluster, like the raspberries you can see, and they hit we call it superheater. So it could be between 500 to 1,000 times hitting better than currently clinically used for phase 3 clinical trial by the company McForce. So then the next thing is that you can see that in order to, can I go to the slide next? Who controls the slide? Okay, so the next thing is that with the nanoparticle, first thing you had to make it biocompatible so it's safe into the body. And the next challenge is 1 in 2 have cancer in their lifetime, so you need tons of these materials. So it has to be inexpensive. So iron is very cheap, and it has to be reproducible, the process, because every time you inject something, you know exactly the dose. It's not like every time you have a 5 milligram or 10 milligram because of variation. And you need to have a green process, and it needs to be scalable because, as I said, that we need tons of these materials and in sustainable reduction because we have to care about our climate. Can I have the next one, please? And then, so the next thing you need is you need to have an alternating magnetic field and the device. So this is the device currently used for clinical trial for the pancreatic cancer, and it was done in Barcelona. And if you come to our stand, you can see that the device very small, less than your palm of your hand, and it already within 10 seconds it can heat up locally the magnetic nanoparticles. Can I have the next one? So to moving forward, we not only use the magnetic nanoparticle alone to kill cancer, but we can have a synergistic effect. That means cooperative effect with chemotherapy. So you can conjugate the drug and then we use together with magnetic nanoparticle. And cancer cell is more acidic. So only when this nanoparticle gets inside the cancer cell, it will release the drug. So it, again, another one more focused to the cancer cell and kinder to the patient. And then the next thing, you can see here that if we're going to have a polymer that responsive to the temperature, like when the magnetic nanoparticle heating up, it can release the drug. So we also have a thermal-responsive polymer demonstration downstairs. So that is how it works. Thank you.
Wow, okay. Super complicated, but very well exampled through the gingerbread man. And I just wonder, Professor Forster, you sort of work more directly with patients. What sort of difference will this make to them?
Well, I think we know that, I think in general we know that the first treatment we ever had for cancer was surgery. As Professor Bryce was saying, if you found a lump of something, you chopped it out. But depending on where it's located, unfortunately, that can often cause damage to the other parts of the body nearby. And they're damages which you have forevermore. So even if you have a successful treatment that might cure the cancer, the complications are the poor person has to live with the consequences forevermore. So a lot of the treatments we're talking about, in fact, both of the technologies we're talking about really are looking at trying to maintain the ability to cure and get rid of the cancer, but cause less damage to the normal cells around it. And so the idea with this is if you can inject into the tumour the particles, they're going to heat up and kill the cancer cells, have less damage to the surrounding cells, and then in the next generation, if you can attach. So I spend most of my day injecting or giving people different drug treatments for their cancer, which have a good effect on the cancer, but unfortunately go around the body and treat and hit other parts of the body as well. If you can have that effect but more localized to the tumour, it's released only in the tumour and not elsewhere, you get more bang for your buck, essentially. You're getting all that damage to that area and less damage elsewhere. So what we're hoping is that these sorts of technologies will allow us to have the same sort of an effect, but with less side effects.
And how far off do you think this technology is, all being well?
Well, it's a slow process, unfortunately, and I suppose that's good in a way because we need to make sure all of these treatments are safe for patients before we use them. But there's a lot of long years of work has already happened in the sort of development of the idea and the proof that that idea works in a test tube or in different model systems.
How long has it taken you to get to this point?
To this point now, Thanh, you've been doing this for
5 years.
5 years.
20 years.
20 years. 20 years. Okay. Wow. Okay. So how long until?
And then I think once it goes into a person, often we've tried to accelerate the evaluation of these new technologies in patients. It used to be that it would take probably a decade. We're now trying to shrink that up so that we can get results within. Certainly still takes 4 or 5 years because you have to first of all show that the new treatment's safe. Then you have to work out how much you want to give because you want to get maximum effect without causing damage. So there's a bit of evaluation there which takes a bit of time. And then you want to show, well, actually, we've got other treatments. Is this better than our normal treatments? So then you have to do a trial which shows, is this better than what we normally do? And those studies tend to be bigger, involving more people, and they take time. And of course, if you're in a situation like this where you're looking at trying to cure a cancer, then you don't get an answer straight away. You will need to see, actually, does the cancer stay away for a long time? So you need to observe the people for quite a long time afterwards. So the process is quite long. Yeah. It takes a number of years. But during that time, people can sometimes access this treatment through the clinical trials. So it's not sitting in the shelf. Once it gets into the clinic, people are accessing it, but within the context of a controlled trial, so it's safe and it's regulated and monitored carefully.
And have you started the trials yet, or are they still?
Not quite. Well, not with this particular technology. So there are these magnetic particles in clinical trials, but this particular flower is hopefully within the next couple of years will be coming into the clinic, but not quite yet.
A couple of years. Okay, well that's good. And what about this side in terms of your particle beams? When do you think that might be being used?
We've laid out a timeline. We have to do the research to get to the point where we can propose a clinical facility. The key thing that we're trying to do is to do this as a collaboration. So the LARA collaboration actually involves clinical oncologists all the way to laser plasma theorists. The point is that we want to create an activity where we are able to deliver benefit straight away. So an example would be that our industrial partner, Leo Cancer Care, so if you visit the stands, you'll find their upright patient positioning system. Things that we will need for the final system, in principle, we can use today in things which would be in the clinic and would give benefit in the clinic. One example is automatic patient positioning, automatic dose verification. Other examples would include the biological measurements that we want to make.